Hello friends and welcome to CEC online lectures. In my previous lecture on the plant hormone auxin, I gave a brief overview about the discovery of auxins, followed by their biosynthesis in plants. We also discussed how they are transported in plants, and in that we discussed that auxins have a polar movement, that is they move from the SAM to RAM. SAM is shoot apical meristem and RAM is root apical meristem. We also discussed that if we see the movement of auxins in the shoot, it is normally basipetal, that is from the tip to the base, and in roots it is acropetal, that is from the base of the roots to the apex of the root. We also discussed about some of the physiological actions exhibited by auxins and their mechanism of action. So friends, today I'll discuss about another class of plant hormone, that is gibberellins. Now it all began in 1898 when Konishi, who was a Japanese farmer, he dictated an agricultural book because he was a semi-literate person. So he dictated a book in which he gave the description of a very common rice disease, which we now called as Bacchanae disease or the foolish seedling disease. And he described some of the most important characters of that particular disease, that is appearance of tall or spindly plants and their yellowing of the leaves. Now from 1898 to 1910, Hori, who was a scientist in Japan, worked on this disease and gave its proper description and also the causative agent of this disease. And according to him, fungus was the major reason or the major causative agent of this disease, though he was not able to identify the fungus. Later in 1926, it was Kurosawa who showed that the disease, Bacchanae disease, is caused due to the fungus Gibberella jujicori, when he was studying on this fungus in rice and the maize seedlings. Though in 1931, Wollen Weber, another scientist, who established that Bacchanae is caused by the Fusarium moniliform, which is the asexual or the imperfect stage of Gibberella. But by and large, most of the literature shows, and they considered Bacchanae disease is caused by the Gibberella fungus. Later in 1935, Yubota, another scientist from the Tokyo University, he started his research on this fungus in order to identify the causative agent that is present in the fungus. So he prepares certain filtrates or the extracts from the fungus and he named the causative agent as gibberellin. So this is how the term gibberellin was originated, and Yubota was the person who gave the term gibberellin. Later in 1938, Yubota and his co-worker Sumuki, they announced the isolation of two crystalline substances from the fungus, and these substances were named as gibberellins A and B. Now interestingly, the Western world was completely unaware of gibberellins until 1950, when Yubota visited Europe and he presented certain Japanese papers that were related to gibberellins to the Western world. And after that only the Western world start working in the field of gibberellins. And in 1955, Stodola, who was a scientist in US Department of Agriculture, started purification of gibberellins from the fungal extracts, and a new compound which he named as gibberellin X was isolated. Later on, this gibberellin X was given the name as gibberellic acid or GA3. In 1956, West and Finney in the University of California, and along with Margaret Redley from the Acres Research Laboratories in England, they discovered gibberellins from the higher plants, specifically from the seeds and fruits of the plant that belong to 9 genera representing 7 families of angiosperms. So this was the first instance when the gibberellins were isolated from the higher plants. In 1965, Plague gave the first definition of gibberellins, and according to him, these are the compounds having an end-gibberellin skeleton and biological activity in stimulating cell division or cell elongation or both or such other biological activities. So as you can see in the picture, the end gibberellin structure is a tetracyclic structure having 4 rings A, B, C and D. Another definition was given for gibberellins pertaining to their chemical structure or chemical nature. And according to this definition, gibberellins are defined as naturally occurring tetracyclic diterpenoid acids with structures that are based on the end gibberellin carbon skeleton, and they are generally named as Ga followed by the number in the chronological order of their identification. Say for example, the first gibberellin that was isolated was given name Ga1, followed gibberellin was given the name Ga2, and eventually Ga3, Ga4, Ga5 in order of their discovery. And till date, more than 130 gibberellins have been identified from more than 120 plants and around 7 bacteria and 7 fungi species. Now, gibberellins, depending upon their chemical forms, can be categorized into 3 major categories: free gibberellins, conjugated gibberellins, and the water soluble or the bound gibberellins. Now, the first 2 classes, that is free gibberellins and conjugated gibberellins, are very well chemically defined, whereas the third category, that is water soluble or the bound gibberellins, have not been defined well chemically, and therefore the third category is considered in a very limited manner. Now, various structure activity related studies have also been performed on gibberellins, wherein what are the structural components that are responsible for the activity of gibberellins have been identified. And according to these studies, as I discussed, that gibberellins are the tetracyclic structures. So all the 4 rings that are present in the gibberellin structure are essential for the potent action of the gibberellins. Or in simple words, if I say, then all the 4 rings of the gibberellin structure are required for the activity of gibberellins. Then there should be a carboxyl group present at the 6th carbon of these compounds. And thus the carboxyl group at 6th position is also essential for the activity. It has further also been observed that most active gibberellins which have been tested till date, they possess a lactone ring within the ring A of the structure, and this lactone ring is formed between the carboxyl group that is present at C4 and the 10th carbon that is present between the ring A and B, as you can see in the structure. Now, gibberellins have also been classified on the basis of the number of the carbon atoms that are present in their structure. And according to this, they are categorized into C19 gibberellins and C20 gibberellins. Now, it has been observed that the C19 gibberellins are more active than the C20 gibberellins, and C20 gibberellins are also sometimes considered as the precursors of C19 gibberellins, and as we will discuss further in their biosynthetic pathway. Now, gibberellins that possess the highest biological activity or the most active gibberellins amongst those more than 130 identified from the nature: gibberellin A1, A3, gibberellin A4, gibberellin A5 or GA6 or GA7 have been found to be the active gibberellins. And out of these 6 gibberellins, GA1, GA3, GA4 and GA7 are considered more active. And amongst these more active 4 forms, GA3 is the most active gibberellin that has been found in the higher plants. Now we move on to the biosynthesis of gibberellins. Now, the biosynthetic pathway or the biosynthesis of gibberellin takes place in the 3 parts or the 3 components of the cell. The first part of the synthesis takes place in the plastids, which leads to the synthesis of a tetracyclic hydrocarbon known as ent-corene, as you can see in the biosynthetic pathway. And this ent-corene is one of the most important intermediate in the biosynthesis of gibberellins. Now, this ent-corene further undergoes certain structural modifications, and then it moves into the second region of the cell, that is the endoplasmic reticulum. Now the second part of the biosynthesis occur in endoplasmic reticulum, where entchorin subsequently undergo oxidation reaction, and it give rise to the first formed gibberellin, which is known as GA12, and its 13 hydroxylated analog, which is known as GA53. Now GA12 and GA53 then enter into the third part or the third area of the cell, that is the systole. And the third part of this biosynthesis takes place in systole. And in systole, GA12 and GA53 are further oxidized, and they undergo various structural modifications with the help of various enzymes to give rise to C20 gibberellins, and these C20 gibberellins further give rise to C19 gibberellins. Now the synthesis or the biosynthesis of gibberellins, it largely occur in the cells of young leaves and apical buds. It has also been found to occur in the developing seeds and fruits, and in the apical region of the roots. Now we move on to the transportation of gibberellins. So as we discussed in case of auxins, the transportation of auxins was polar. But unlike auxins, gibberellins have a non-polar movement. That is, they do not move from SAM to RAM, that is shoot apical meristem, to the root apical meristem. On the contrast, gibberellins have a bidirectional movement, because we discussed in my previous lecture that auxins have a unidirectional movement, that is from shoot to the root, shoot apex to the root apex. But gibberellins have a bidirectional movement, that is they can move both from shoot to root as well as from root to shoot. So when they are moving from shoot to root, this movement occurs through the phloem tissue, and when they are moving from root to shoot, this movement occur, or this transportation occur through the xylem tissue. Now we move on to the physiological actions of gibberellins. Now, as I told you in the definition, that these are the compounds which are responsible for the cell division or the cell elongation. So one of the prominent action of gibberellins is the cell elongation, and this effect is mostly seen in the internode region of the plant, and thus we can say that gibberellins also cause the internode elongation, and therefore these gibberellins are helpful in removing dwarfism of the plant. That is, they help in overcoming the short height of the plant in some of the plants. Now this response of cell elongation that is exhibited by gibberellins, this occur in the same manner as we discussed in the case of auxin. That is, the gibberellins also cause elongation by loosening of the cell wall by activation of the enzyme that causes the breakdown of bonds of xyloglucan, which is an important hemicellulosic component in the cell walls. And once these walls are break, the cell walls are loosened, which leads to the elongation of the cell. Now there is a difference between the action of gibberellins and auxins here. In auxins we discussed that once these cell walls were loosen up or they become elastic, simultaneously there was a movement of water inside the cell, which increases the turgor pressure, which leads to the elongation of the cell wall. But in case of gibberellin, there is no increase in the turgor as we observed in the case of auxin. So this is the basic difference, how the gibberellins cause cell elongation and auxins causes the cell elongation. Now further, it has also been observed that gibberellins cause degradation of the repressor proteins, which are known as the DELA proteins. So as you can see in the image, now these DELA proteins are the repressor proteins, means they causes the inhibition of the growth of the plant. Now these proteins are also considered as nuclear transcriptional regulators, and they also inhibit the gibberellin signaling, thereby restricting the plant growth. And this restriction is perhaps caused by the transcriptional reprogramming that is exhibited by these DELA proteins. Now DELA proteins contain two major domains. First one is a conserved carboxy terminal, or also known as GRAS domain, GRAS, which is responsible for the transcriptional regulator function. And the second one is an amino domain or the terminal, which is required for the gibberellin induced degradation. And how this happens, we will discuss it further. Now, when gibberellins are applied, or after their biosynthesis, when they reach to the target site, they bind to the GA receptor, which are known as GID1 or the GID1 receptors, which are nothing but the gibberellin insensitive dwarf 1 receptors. Gibberellin insensitive dwarf 1 receptors. Now, these GID1 receptors, they also contain two major domains. First one is the carboxy domain or the carboxy terminal that forms the GA binding site or the pocket. That is, this is the region carboxy terminal. This is the region where the gibberellin binds to the receptor. And the second one is an amino terminal extension domain, which is also known as NX domain. And this domain act as a lid for the region where gibberellins are bind. Now, the binding of gibberellins to the carboxy terminal domain or the carboxy terminal of the GID1 receptor induces a conformational change in the structure of the NX domain. And this conformational switch causes the movement of NX domain over the GA binding region or the GA binding pocket, as if a lid closes that particular pocket, as you can see in the picture. And further, it also creates a hydrophobic surface which attracts the DELA protein, and therefore it acts as the site for the DELA protein binding. Though there is no contact between DELA and the gibberellin, there is no direct contact between DELA and gibberellin, because in between the two there is the NX region of the GID1 receptor. But still, the binding of the DELA to this GA or the gibberellin GID complex further stabilizes the overall complex of DELA, gibberellin and GID1 receptor. So we call this whole complex as gibberellin, GID1 and DELA complex. Now, once this complex is formed, it further bring certain conformational changes in the rest of the DELA protein structure, and these conformational changes make DELA protein more prone towards a specific type of protein which is known as F-box protein. And these F-box proteins promote polyubiquitination of DELA through SCF SL1 GID2 ubiquitin E3 ligase complex. So in simple words, F-box proteins further increase the ubiquitination of the DELA protein through SCF proteins. And this ubiquitination of DELA protein causes their degradation. And once the DELA proteins are degraded, the inhibitory effect of DELA protein on gibberellin is removed, and thereby this causes the restoration of the growth response in the plants. So this is how gibberellins show their growth effect on the plants. Now, according to another hypothesis, because it has been observed that auxins also increase the length of the cells, there is another hypothesis which state that since auxins increase the gibberellin production, the increased length of the cells is also due to the increased GA production, and it is not only because of the auxins. So this is another hypothesis. Now in this picture you can see the relation of gibberellins with other plant hormones and how other sources impact the biosynthesis of gibberellins. So as you can see that auxins, they increases the synthesis of gibberellins. They have an impact on the increased concentration. They basically increase the concentration of gibberellins, whereas ethylene and cytokinins along with abscisic acid, they basically inhibit the production of gibberellins. Light during the germination stage, it enhances the production of gibberellins, whereas light in the hypocotyle region, it decreases the gibberellin production. Certain biotic stresses, they increase the gibberellin production. Certain abiotic stresses like cold and salinity, they decrease or inhibit the gibberellin action or decrease the gibberellin production. Now, as we discussed that gibberellin binds to GID1 protein and thereby inhibit the DELA proteins. Now, as you can see that DELA has inhibitory action on various proteins. First one is PIF protein, PIF3 and PIF4. Now, these PIF proteins are responsible for the elongation of hypocotyle, and these PIF proteins are inhibited by DELA protein. Another protein, SCL3 protein, it is also inhibited by DELA and it is responsible for the root elongation. So DELA inhibits SCL protein, thereby inhibiting the root elongation and seed germination. DELA also inhibits ALC protein, which inhibits the development of valve margin. It also inhibits the JAZ proteins, which inhibit the JAZ mediated defense responses. And DELA has also been found to inhibit other gibberellin induced responses, though the mechanism is not clearly understood. So as we discussed, gibberellins bind to GID1 receptors, which eventually inhibits the DELA protein, thereby removing the inhibitory effect of DELA on PIF protein. And since now the PIF protein will become active, they will cause hypocotyle elongation. Similarly, SCL3 proteins will also get active and they will cause root elongation and seed germination. And all those effects that were inhibited by DELA are reversed by gibberellins, thereby causing increased plant growth. So this is a broad overview that how gibberellin interact with other hormones and how they exhibit their growth effect on the plant. Now, gibberellins also enhances the cell division, another physiological action, but this enhancement is not primary reason for the growth. Now, gibberellins increase the expression of genes that cause stimulation of cyclin-dependent protein kinase, that is CDKs, and M cyclins, which eventually result in the enhanced cell division, mainly between G2 and the M phases of the cell cycle. Gibberellins also stimulate the germination of seeds. Now, it has been observed that once gibberellins are produced in embryo, they diffuses into the aileron layer of the seeds and promote the expression of MYB transcription factor, which is also known as GA-MYB factor because it is associated with gibberellins, therefore GA-MYB factor, which further binds to some promoters of alpha amylase, thereby increasing the expression of this enzyme. Now, it has also been observed that gibberellins also promote the alpha amylase mRNA production. Now, this increased expression of the enzyme or increased production of the enzyme causes the hydrolysis of the starch, because we know that alpha amylase is the enzyme which is responsible for hydrolysis of starch, and it breaks down the starch, which is the main food source in the endosperm, and thus it provides sufficient nutrient for germination. Thereby, this is basically how gibberellins exhibit or stimulate the germination of the seeds. Now, gibberellins also promote the leaf expansion and root elongation, but it has also been observed that at higher concentration they inhibit the root growth. Therefore, it is said that gibberellins can change the root to shoot ratio. Now, gibberellins also stimulate flowering in the plants, but this effect varies from species to species and also is dependent on the light conditions, because it has been seen that in some plants long photoperiod shown to induce flowering when GA is applied, and in some plants it is not observed. Now, GA also help in change in the gender expression of a flower, and it has been found they help in increasing the maleness of the dioecious flowers of a particular plant. Now, gibberellins also cause setting of the fruits and their growth. A number of dioecious fruit trees such as apple, pear and some citrus species have been found to get benefited from the application of gibberellins, as they help in inducing or in setting up the fruit, as well as it has also been found to increase the size of the fruit whenever gibberellins are applied, and whenever gibberellins are applied in combination with auxins. Now, gibberellins are also used extensively for inducing parthenocarpic fruits, that is the seedless fruits, specifically in case of grapes. As I already told you that gibberellins also help in increasing the size of certain fruits, and grapes is one of the examples. So friends, this was all about gibberellins, and in my next lecture I'll discuss about cytokinins, which is another class of plant hormone. Thank you.